Hello children, last time we had started with the topic on catalysis. Catalysts were those substances which could increase the rate of the chemical reaction, isn't it? That was what we had seen in the previous class. We had learnt about the different types of catalyst, heterogeneous catalyst, homogeneous catalyst. We had seen that enzymes were one of the catalysts which were involved in the biochemical reactions occurring around us. So, we had studied about enzyme as the catalyst. Today, we will be studying about the characteristics of enzyme, how enzymes act as a very good biocatalyst. What are their characteristics? What are their significance? That we will be learning about today. Okay? So, one by one we will take all the different characteristics of enzyme. First characteristic of the enzyme is specificity. Now, what do you mean by specificity? Means every catalyst is specific for a particular reaction. It means that one catalyst will not be able to catalyze many number of reactions. It will be able to catalyze only one kind of reaction. For example, see, we will take the example of urease. Urease is a catalyst which will decompose or hydrolyze urea, NH2CO, NH2, this is your urea. This urea will get hydrolyzed in presence of urease to give you ammonia and CO2. Okay. But this urea will not be helpful to catalyze the reaction of N-methyl urea into a reaction. It will not cause the reaction to occur. So, here if you are using urease enzyme to catalyze N-methyl urea, then it will give you no reaction. It indicates what? Ki this urease enzyme, it is specific for this particular reaction only, where it will catalyze the urea and give you the product as ammonia and carbon dioxide. Okay? So, it is specific for this reaction. Similarly, you can take another example like say C12H22O11. What is this compound C12H22O11? This is known as sucrose. Okay? This sucrose, when you hydrolyze in presence of the enzyme, invertase, okay, in the presence of enzyme invertase, it will give you the molecule glucose C12H22O6 plus C6H12O6 C6H12O6. This is the molecule of glucose and this is the molecule of fructose. ठीक है, तो यहाँ पे ये जो इन्वर्टेज एंजाइम है वो स्पेसिफिक होता है टू कन्वर्ट द सुक्रोज इनटू द मॉलिक्यूल्स ऑफ ग्लूकोज एंड फ्रुक्टोज. ओके, द सेम एंजाइम इन्वर्टेज will not be able to hydrolyze maltose. Again, that is also represented by the same molecular formula, but it will have a different structure. Okay, so maltose को hydrolyze नहीं कर पाएगा invertase. It will be hydrolyzed by another enzyme known as maltase into molecules of glucose C6H12O6, 2 molecules of glucose. So, do you understand the meaning of specificity now? That every enzyme is specific for a particular reaction. Urea will be hydrolyzed only by the enzyme urease. Sucrose will be hydrolyzed to give you glucose and fructose only by this enzyme invertase. Maltose will be hydrolyzed only in presence of maltase enzymes to give you glucose. Similarly, glucose again can be converted to ethanol only under the presence of zymase enzyme. So therefore, these enzymes, they are specific for a particular reaction. That is what we call as specificity of enzyme. A particular reaction won't be a particular enzyme will be able to hydrolyze or carry out the reaction of one kind only. It will not be able to carry out the reaction of other kind. That is what is called as specificity. Clear now? So that is specificity of the enzyme. Second is efficiency. Efficiency means the enzymes, they are efficient catalysts and they speed up the reaction by 10 power 20 times the normal reaction. Okay, इसमें भी हम एक example लेके समझ सकते हैं. Say for example, amide. An amide of any kind. It requires heating for the reaction to occur. This is in the laboratory condition. Okay. In the laboratory condition, for any reaction of an amide to be carried out, heating is required for at least 2 to 3 hours. Then only this reaction will proceed. Any kind of reaction which we have to carry out with amide. For the decomposition of amide, heating is required for 2 to 3 hours. But the same amide which is present in protein in our body, the protein molecules are present in our body and the proteins are being digested in our body. But those proteins, they are easily catalyzed with the help of enzymes like peptase enzyme. है ना? ये जो proteins हमारे body में present है, we are having the intake of proteins. Those proteins also contain amides, but they are very easily digested within few period of time with the help of biocatalyst like enzymes which are present in our body, in the stomach. The protein is being digested in the stomach itself, isn't it? So therefore, proteins are being digested in the intestine. So there the particular enzyme, it is very making it very easy for the amides to get hydrolyzed easily. Are you understanding? So therefore, this is what we call as efficiency of enzymes. Normally the reaction which takes place at very long period of time, these enzymes make that reaction very easy and in less period of time. Okay? So that is what we call as efficiency of enzyme. जैसे amides है in normal condition in laboratory, it will take 4 to 5 hours for the reaction to have this amide decomposed. So, but proteins, okay, proteins which contain amides, are easily decomposed or digested by the enzymes. ठीक है? So that is what we understand by efficiency of the enzymes. The third point is key, the enzymes, they are required only in small quantity, very less quantity. It is not required in large amount. Why? Because as soon as the reaction is over, the catalyst is regenerated and that catalyst is now ready for the fresh reactions between the reactants. So therefore, very small quantity of the catalyst is enough for a particular reaction to occur. एक example ले लेते हैं हम, देखो, a single molecule of carbonic anhydrase can decompose 36 millions of molecules of H2CO3 to CO2 and H2O just in 1 minute. So you can see only one molecule, it can carry out the reaction of 36 million molecules. of H2CO3. If I write the reaction, it will be this way. See, H2CO3 carbonic anhydrase. This is carbonic acid, okay, carbonic anhydrase. ये होता है उसका particular enzyme to convert the carbonic acid into H2O and CO2 just in 1 minute, okay. And also you can see that the reverse reaction, it occurs in presence of carbonic hydrase, okay. That also very less amount, you can have this ratio 1:36. One molecule of catalyst will be able to catalyze 36 million of molecules of H2CO3 and vice versa, that is CO2 plus H2O giving you H2CO3. So, that is the meaning of small quantity. That is the characteristic of enzyme, ki enzyme only very little amount of enzyme is required to catalyze a reaction. Followed? Okay. So, you can note these points, then we can go to the next point. The next characteristics of enzyme is optimum temperature and pH. Optimum temperature means the maximum temperature at which the enzyme can work effectively, that is called as the optimum temperature, and that temperature in our body it is 37 degree centigrade, okay. And pH range in which the enzymes can be active in proper manner, it is pH equal to 7.4. So, this 7.4 pH and temperature 37 degree centigrade is called as the optimum temperature and pH in which the enzyme can work properly, okay, appropriately. That is called as the optimum temperature and pH. The next point which you come across is enzyme activators. Enzyme activators, or we call them as coenzymes. Now, if some vitamins are present along with the proteins, no, it will enhance the activity of that enzyme, okay. So, these are all ions which are known as coenzymes, which help the enzyme to increase its efficiency. जैसे Na plus है, Mn plus 2 है, Cu plus 2, Co plus 2, they are all known as coenzymes. These are ions which increase the activity of the enzymes. So, therefore, they are called as either enzyme activators or it is called as coenzymes. ठीक है? For example, see the activity of enzyme amylase. Amylase is the enzyme which converts starch into glucose. So, that enzyme ka activity is increased by the presence of NaCl, which provides Na plus ions. So, here Na plus ion is the coenzyme for the enzyme amylase. Have you understood? So, enzymes का activity को increase करने के लिए there should be some ions, or there are ions which are present in our body. So, those ions are called as coenzymes. ठीक है? उसी तरह से enzyme inhibitor भी होता है. This was enzyme activator, enzyme inhibitor. Enzyme inhibitor means it decreases the efficiency of the enzyme, or it acts as a poison to the enzyme. That means it will not allow the enzyme to carry out its function properly. जैसे a certain enzyme is causing is making a reaction occur in your body, okay, and that reaction is causing harm to your body. So, in that case, when you take a medicine, those medicines will act as a inhibitor for that enzyme, or will act or will not make that enzyme to work, because that particular reaction is causing some harm to your body. So, in that cases, those substances which decrease the activity of the enzyme, that will be called as the enzyme inhibitor, okay, or it may be called as the poisons. So, these were the different characteristics of enzymes, okay. I hope you have followed that. Enzymes are the biocatalysts which are carrying out various biochemical reactions in your body, and these are the different characteristics of the enzyme which make the biochemical reactions effective. Okay, so note this. Now we will study about how the enzymes work, that is mechanism of enzyme catalysis. ठीक है, ये जो मॉडल है, this is known as the lock and key model. Just as every lock will be opened only by a particular key, similarly enzyme is also specific for a particular reaction. A particular enzyme will be able to catalyze only a kind of reaction. Another reaction won't be able to be catalyzed by this particular enzyme. So it is specific for a particular reaction. That is what that is the characteristics of the enzyme. Okay. Now let us see how these enzymes undergo the catalysis process. Usme here I have given a representation of an enzyme here. Okay. This enzyme, it has a region on it which is called as the active sites. What is it called? It is called as active sites. देखो ये जो region है उसका एक specific shape होता है और वो specific shape को हम कहते हैं active site. So it is at this site the reactant molecule will get itself attached to it. Okay, तो ये जो reactant molecule है, it is known as the substrate. Okay, this substrate molecule will be able to bond itself with the enzyme at the active sites. और ये जो enzyme का active sites है ना, वहाँ पे there will be functional groups like NH2 group or CO group or any other functional groups will be present. A particular functional group will be present. Okay, and that particular functional group will have an attraction for the reactant molecule. Okay, तो ये जो reactant molecule, a particular reactant molecule will only be able to attach itself to this enzyme क्योंकि वो active site is such that it will have a particular shape और वो shape में सिर्फ यही particular reactant molecule fit in हो सकता है. So that is how only this reactant molecule will be able to attach itself with the enzyme, and this attachment takes place through some weak bonds like hydrogen bond or van der Waals force of attraction. तो ये दोनों इस तरह से there will be a bond formation taking place, and now you have a enzyme substrate complex. Okay. So this substrate or the reactant molecule have got attached to the enzyme at the active sites by van der Waals force of attraction or hydrogen bonding. ठीक है, it forms an enzyme substrate complex. Okay, that enzyme substrate complex will now get converted into enzyme product association. The reactant molecules will associate itself, will come together, will undergo reaction, and it will get converted into product. So now the enzyme product association you can see here. This is your enzyme, this is your product. Okay, so enzyme product association you can observe here, and this product will then have the tendency to detach itself from the enzyme, and you have got your product. So this is basically how the enzyme catalysis works. Okay. The things to remember is that each enzyme, it has a particular active site on which the substrate molecule gets itself attached. Okay, और वो specific होता है, only this particular reactant molecule or substrate will be able to attach itself to that enzyme. तो इसीलिए enzymes जो होते हैं वो specific होते हैं for a particular reaction. ठीक है, it will get converted to enzyme substrate complex. The complex will now be having the product in it. So it now will be called as enzyme product association, and then the product will detach from the enzyme. Okay, the desorption will take place. So this is what we understand as lock key model for the enzyme catalysis. Okay, so you can note it, already it is there in your textbook, so you can take it from there also. One more thing you can note down is कि ये जो the attachment of the substrate with the enzyme, ये जो reaction है, this is a fast reaction. Also it is irreversible. Okay. While the next reaction, that is formation of the product, it will be a rate slow. So therefore it is, this will be a slow process. So therefore this will be a rate determining, it is rate determining step. Okay. So formation of the enzyme substrate complex, it is a fast reaction, while the formation of enzyme product association, that is for the reaction to occur, the reactant molecule should get converted into product. No, that will be a slower process, and therefore it will be called as rate determining step. Okay. So you can note this down. Some of the industrial catalytic processes, it is given in page number 135 of your textbook. See, the processes like Haber's process. Haber's process is used for the manufacture of ammonia, and that manufacture of ammonia is carried out in the presence of finely divided iron, which is acting as the catalyst, and molybdenum is used as a promoter. Okay. At this using this catalyst, the conditions of the reaction can be made less vigorous, that is the reaction can be carried out at the optimum pressure of 200 bar pressure and 723 to 773 Kelvin temperature. Okay. Nowadays it is also seen that instead of finely divided iron, we are using iron oxide. So in many books you will find either the catalyst being written as Fe, that is iron, or FeO, which is iron oxide. Okay. The second process in the industry where you use the catalyst is in the Ostwald's process for manufacture of nitric acid. So that is catalyst used in that case is platinized asbestos. Okay. That is taken at a temperature of 573 Kelvin. Then contact process you have learnt when we were learning about p block elements. So contact process is used for the production of H2SO4 or sulfuric acid. For the conversion of sulfur dioxide into sulfur trioxide, the catalyst which was used in contact process was vanadium pentoxide, that is V2O5, and this vanadium pentoxide is used at a temperature of 673 Kelvin to 723 Kelvin. Similarly, for ion chamber process, the production of H2SO4 is carried out in the presence of NO as the catalyst. Okay. That also we had discussed previously. So, these are some catalysts which are used in industries. Only few are given, there are many. We have seen that hydrogenation reactions where you are obtaining vegetable ghee from the vegetable oil, that is using nickel as the catalyst. So, various other processes are there where you are using catalyst to increase the rate of the reaction. Few are given here that you can go through. Okay. So, this completes the topic on catalysis. One small topic on colloids is remaining, that is calculation of gold number. So, that we will be doing in the next section. Okay. This topic is based on protective action of the lyophilic colloid. You had seen there were two different kind of colloids based on the interaction between the dispersed phase particle and the dispersion medium, isn't it? One was lyophilic colloid, another was lyophobic colloid. Lyophilic colloids, they are quite stable because of a covering of the dispersion medium particles around it. But lyophobic colloids जो होते हैं वो unstable होते हैं. So, these lyophilic colloids are added to the lyophobic colloids so that they can become stable. See, if suppose you are representing this particle as the lyophobic particle, okay, This is the lyophobic particle. Okay, it is unstable in nature. The lyophobic salts, they are unstable, isn't it? So why are they unstable? Because they have the tendency to aggregate together and get converted to precipitate form. तो ये लायोफोबिक कोलॉइड्स को हम अगर स्टेबल करना चाहे तो वी आर एडिंग अ लिटिल अमाउंट ऑफ लायोफिलिक कोलॉइड इनटू इट. Okay, so those lyophilic colloid particles, it will surround the lyophobic colloid particles and make it the attraction between the two particles is disturbed here, because these lyophilic particles will not allow the lyophobic particles to come in contact with another particle of the lyophobic particle, है ना. तो ये जो lyophilic colloid हम जो add करते हैं, that is called as the protective colloid. What is it called as? It is called as the protective colloid. ठीक है. So this is the lyophilic particle. Okay, and this lyophilic particle is acting as a protection to the lyophobic particle, which prevents it from the aggregation. ठीक है. So that is why these lyophilic particles are called as the protective colloid. Okay. Now, to compare the protective action of these lyophilic colloids or colloidal particles, a number was introduced which was known as the gold number. How is it defined? Gold number is the gold number of a protective colloid. It is the minimum mass of it in milligrams which must be added to 10 ml of standard red gold solution, so that no coagulation of the gold take place, that is gold salt take place, when 1 ml of 10% NaO solution, 10 ml of NaCl solution is rapidly added to it. So what is the definition? Once more we will read it out. Gold number of the protective colloid is the minimum amount of the substance which should be added to 10 ml of standard red gold solution, so that no coagulation of the gold solution take place. No coagulation of the gold salt should take place. That is, it should not convert from red color to blue color when 1 ml of 10% solution of NaCl is rapidly added to it. Okay. So when you are adding NaCl which is 10%, how much solution you are adding? 1 ml of NaCl when you add to the red gold salt, then that salt should not get coagulated. Okay. That much amount of the protective colloid which should be added, that amount of colloid is known as the gold number. Okay. So that is the definition of gold number, and we have found experimentally that smaller the gold number of the protective colloid, then greater is its protective action. Okay. I hope you understand what is gold number. It is a minimum amount of the protective colloid which can be added to a gold solution, which we call it as standard red gold solution. How much ml of gold solution we are taking? 10 ml of gold solution. ये 10 ml of gold solution में हम कितना protective colloid डाल सकते हैं? Minimum amount, so that the coagulation should not take place when 1 ml of 10% NaCl solution is added to it. That amount of the protective colloid is called as the gold number, and it should be in terms of milligrams. Remember that. We will do a question based on it so that you can understand it properly. Okay? So you can note the definition, everyone. Now let us do a problem to calculate the gold number for starch. According to the definition, what was the gold number? The gold number was the amount of the protective colloid which is added to the 10 ml of gold solution or gold salt so that it does not get coagulated when 10 ml of 10% NaCl is added. This was the definition. So, according to the definition, we will try to find out what is the gold number for the starch. Now, in this problem, what is given? The coagulation of 100 ml of colloidal salt of gold is completely prevented by addition of 0.25 gram of starch. You are asked to find the gold number of the starch. Okay. So, for 100 ml of gold salt, to prevent its coagulation, how much is the amount of starch added? That is 0.25 gram of starch is added to prevent coagulation. So, 0.25 gram, that means it is 250 milligram. Okay. So, for prevention of coagulation of 100 ml gold salt, 250 mg of starch is added. But according to the definition, we calculate gold number for 10 ml of the gold salt. Okay. So, therefore, for 10 ml of gold salt to prevent coagulation, what is the amount of starch that should be added, and that amount will only be called as the gold number. Okay. So, how much mg it will be, how many milligrams it will be. So, therefore, it will be 10 into 250 by 100, okay, that is 25 gram. So, therefore 25 gram of starch should be added to prevent coagulation of 10 ml of gold salt. Okay? Therefore, gold number of starch will be equal to 25 gram. Okay? So, this is how you should be calculating the gold number. Okay? One question I will give you for homework that you can do and find out the gold number for the particular protective colloid. Okay? So, this is how you should be carrying out the numericals. Okay?